All right. My name is Molly Sutherland, and I'm an assistant professor at the University of Delaware in the Department of Biological Sciences. And I'm excited to be here today to tell you about my research on cytochrome C biogenesis in bacteria. So I'm going to start by giving you a little bit of background about what cytochrome C is and why you should care about it. I'm going to tell you a short story about one of the pathways called System 2, and tell you a little bit about the future directions of the labs. So what is cytochrome C? It's a highly conserved protein that's found in nearly all organisms, and it's critical for cellular function. In eukaryotes, it functions in the mitochondria for respiration and also plays a role in apoptosis. In the chloroplast, it functions for photosynthesis. And in bacteria, it functions for both respiration, photosynthesis, as well as detoxification. And cytochrome Cs are unique because they require the covalent attachment of heme for proper folding and function. And heme gives cytochrome C its unique redox properties, which allow it to perform all these various functions. So when I talk about cytochrome C biogenesis, I'm really talking about the covalent attachment of heme to a conserved motif in apocytochrome C. So all cytochrome Cs contain a cysteine XX cysteine histidine motif. And when the cysteine thiols are brought in close proximity to the heme vinyl groups, you get covalent thioether bond formation. And then the histidine coordinates with the heme iron. And while there are hundreds of different cytochrome Cs that are known in nature, from anywhere with one heme group to over 10, all cytochrome C biogenesis is accomplished by three pathways called System 1, 2, and 3. So here I'm showing you the pathways. We're not going to go into detail about them, but what I want to point out is that there are two pathways that are found in bacteria called System 1 and System 2. And then a separate distinct pathway called System 3 that's found in eukaryotic mitochondria. So I'm interested in studying these because cytochrome C biogenesis is a fundamental biological process, and I want to understand the basic biology of how cytochrome Cs are made. Also, if you can see from these diagrams, we have this heme molecule here, and it's being moved across membranes and then attached to a protein. So we're developing these pathways as model systems to study heme trafficking, which is important in many different cellular functions. And then finally, we are proposing that the bacterial holocytochrome C synthetases, which is CCMFH and CCSBA, are potential antibiotic targets. So I'm going to tell you today about our work trying to identify the active site of CCSBA, moving towards showing that it could be an antibiotic target. We have a nice system set up for studying these pathways. So we do everything in recombinant E. coli. These are integral membrane proteins, so they're difficult to work with. But with this E. coli system, we can do functional and biochemical assays. And importantly for our proteins, they co-purify with endogenous heme. So I'm going to tell you today about System 2. It consists of the two proteins, CCSB and A. They're proposed to be a bifunctional enzyme, so they function as a heme transporter as well as the holocytochrome C synthetase, so attaching heme to cytochrome C. And to try and provide evidence that it would be a novel target for antibiotics, we asked the question, where is the CCSB active site? And we focused in on a conserved domain called the WWD domain. This is a tryptophan-rich region that's found in 3 of the different prokaryotic proteins. It was found in 1992 and hypothesized to be important for binding heme and positioning heme vinyl groups for attachment. So to test this hypothesis, we developed a novel technique called cysteine-heme crosslinking. So cysteine and heme have this natural propensity to form a covalent bond when they're in close proximity. So we're trying to exploit this reaction to identify heme binding domains. So we mutated the 18 residues of the WWD domain individually to cysteine. We were able to show that they are all stable. Here we're showing the purification. CCSBA purifies as 2 polypeptides. And then we saw if any of these cysteine mutations retained heme at CCSA, which is where the WWD domain is located. We identified 6 residues shown here that have increased levels of heme at the polypeptide compared to wild type. And here I'm showing you a heme stain, which is just a Western blot from heme. So from these experiments and others, we propose that there's an external heme binding domain in the periplasm of bacteria that consists of the WWD domain and 2 conserved histidine residues. And this is what we think is the active site. We did some protein modeling with Sergey Ovchinnikov and David Baker at the University of Washington. So they used genomic analysis, metagenomic analysis, as well as our experimental data to model heme into the WWD domain. We have the tryptophan residues protecting heme and the histidine residues coordinating the heme iron. What this doesn't show, though, is that CCSBA has a very large periplasmic domain. It makes up about 50% of the protein. We have no idea what it does, but we think it's going to be important for either interacting with cytochrome C or somehow gating the access to the active site. So we've tried a lot of different ways to trap cytochrome C in this complex, and we have been unsuccessful. So we're moving on to a new technique using photoactivatable crosslinking. We've chosen to use PBPA. It can be incorporated at engineered UAG sites in your protein of interest. So we have engineered these into cytochrome C. The sites we have chosen are shown here. Any ones with an asterisk, we've actually gotten PBPA incorporation and shown it by mass spec. So our next step is to do the cross-linking reaction. So expose the proteins to 350 nanometers of light, get the cross-link complex, and use tandem affinity purification to purify and identify where the cross-link has occurred. And we're hoping that by doing this, we can understand how cytochrome C interacts with CCSBA and really define our active site. So why do we think that these holocytochrome C synthases may be novel antibiotic targets? Well, first, these pathways are essential in many bacteria. The active site is outside of the cytoplasm. We know that in bacteria, the CXX-CH recognition site is required for heme attachment, while in the eukaryote, we know that there's a CXX-CH required, but also the N-terminal alpha helix of cytochrome C. So that's an additional about 11 amino acids. So there's some sort of different recognition happening. We know in both of the holocytochrome C synthetases that the WWD is the proposed active site. However, in the eukaryotes, there's no WWD domain and there's no known active site. So there's a very different mechanism for heme attachment in the eukaryotes versus the prokaryotes, suggesting that if we can understand those differences, we will be able to rationally design compounds potentially that would affect the prokaryotic pathways, but not the eukaryotes. So with that, I'd like to thank my lab. Amber is my graduate student. Alicia is a rotation student, and Zobi was an undergraduate who did a lot of bioinformatics on the periplasmic domain this summer. At WashU, my collaborators are Robert Clarence, my postdoc advisor, and Arpita Bose, and then I have startup funding from the University of Delaware. And I'd also like to note that the lab is actively recruiting graduate students, so if you have any students who you think might be interested, please send them my way. And with that, I'll take any questions.